Welcome to our exploration of the male reproductive system. In this video, we'll break down the anatomy and physiology that make reproduction possible in males. Understanding these concepts is crucial not only for academic purposes, but also for recognizing symptoms of reproductive disorders, providing effective patient care, and promoting reproductive health. We'll cover everything from the organs involved to the hormones that regulate their function. Let's begin by understanding why this knowledge matters and how it impacts both personal health and medical practice. Understanding the male reproductive system is more than just memorizing anatomy. It equips you to recognize and address conditions like sexually transmitted infections and reproductive diseases. This knowledge also empowers you to educate others about maintaining a healthy reproductive system. Whether you're a student, a healthcare provider, or simply curious, grasping these concepts can help you make informed decisions about health and wellness. Now, let's dive into the core functions shared by all reproductive systems. Both male and female reproductive systems, though different in structure, share four essential tasks. First, they form specialized cells called gametes, sperm in males and ova in females. Second, they bring these gametes together through sexual intercourse. Third, they combine genetic information from both parents during fertilization, forming a zygote, the very first cell of a new individual. Lastly, they support the development of the fetus and the birth of a baby. These shared functions highlight the remarkable coordination between anatomy and physiology in reproduction. Primary sex organs or gonads are the testes in males and ovaries in females. These organs are homologous, meaning they share a common developmental origin. Gonads produce two vital products: gametes, sperm in males, eggs in females, and sex hormones such as testosterone in males and estrogen and progesterone in females. In addition to the gonads, accessory reproductive organs like ducts, glands, and external genitalia play crucial roles in the reproductive process, ensuring the proper transport and nourishment of gametes. Production of gametes and sex hormones is tightly regulated by the hypothalamic pituitary gonadal (HPG) axis. This system involves a series of hormonal signals between the hypothalamus, the interior pituitary gland, and the testes. Key hormones include GnRH, FSH, LH, testosterone, and inhibin. These hormones interact in a precise sequence to ensure proper reproductive function, from the onset of puberty through adulthood. Let's look closer at how this hormonal axis is activated and maintained. Puberty marks the period when reproductive organs mature and become functional. Before puberty, low levels of sex hormones keep the hypothalamus in check, preventing the release of GnRH. As puberty approaches, the hypothalamus becomes less sensitive to inhibition, releasing GnRH in pulses. This stimulates the interior pituitary to release FSH and LH, which in turn prompt the gonads to produce more sex hormone. This hormonal cascade is what drives the physical and functional changes of puberty. Meiosis is a unique type of cell division that occurs only in the gonads. It's essential for sexual reproduction because it reduces the chromosome number in gametes by half. Without meiosis, fertilization would result in offspring with twice the normal number of chromosomes. This process ensures genetic stability across generations and is fundamental to the creation of sperm and eggs. Understand meiosis, it's important to know some key terms. Sister chromatids are identical copies of a chromosome joined together after DNA replication. Homologous chromosomes are pairs, one from each parent, that carry genes for the same traits. During meiosis, these chromosomes pair up and may exchange genetic material, increasing genetic diversity. These foundational concepts set the stage for understanding how meiosis creates unique gametes. Within homologous pairs, non-sister chromatids can exchange segments of DNA in a process called crossing over. This exchange increases genetic variability among offspring. The diploid number refers to the total number of chromosomes in most body cells, 46 in humans, arranged in 23 pairs. Each pair consists of one chromosome from each parent, ensuring a blend of genetic material in every individual. Gametes are haploid cells, meaning they contain only one member of each homologous chromosome pair, 23 chromosomes in humans. When a sperm and egg fuse during fertilization, they form a diploid zygote with 46 chromosomes. This restoration of the diploid number is essential for normal development and genetic continuity between generations. Meiosis consists of two consecutive nuclear divisions, meiosis I and meiosis II. DNA replication occurs only once before the first division. The end result is four haploid daughter cells, each genetically unique. Each division includes four stages: prophase, metaphase, anaphase, and telophase. This process ensures both genetic diversity and the correct chromosome number in gametes. During interphase, all chromosomes are replicated. In meiosis I, homologous pairs separate, reducing the chromosome number from diploid to haploid, a process called reduction division. In meiosis II, sister chromatids separate, ensuring each daughter cell receives an equal share of chromosomes. This sequence is essential for producing gametes with the correct genetic content. Meiosis I is where chromosome number is reduced. Prophase I features events not seen in mitosis, such as synapsis, where homologous chromosomes pair up to form tetrads, and crossing over, where genetic material is exchanged. These steps are crucial for increasing genetic diversity among offspring. During metaphase I, tetrads line up randomly at the spindle equator, setting the stage for genetic variation. In anaphase I, sister chromatids of one homologous chromosome are separated from those of the other pair. This random alignment and separation further contribute to the genetic uniqueness of each gamete. At the end of meiosis I, each daughter cell has two copies, sister chromatids, of one member of each homologous pair, either maternal or paternal. The cells are now haploid, but each chromosome still consists of two chromatids. This sets the stage for the second meiotic division, where these chromatids will finally separate. Meiosis II closely mirrors mitosis, except that the cells entering this stage are haploid, not diploid, and chromosomes are not replicated beforehand. The result is the separation of sister chromatids, producing four haploid cells, each with a unique genetic makeup. This final step ensures the diversity and proper chromosome numbering gametes. Mitosis ensures that all body cells have the same DNA for growth and repair, producing identical cells. In contrast, meiosis cuts the chromosome number in half and introduces genetic diversity. Random alignment of homologous pairs and crossing over during meiosis ensure that each gamete is genetically unique, which is vital for evolution and adaptation. Before meiosis begins, the cell enters interphase, during which all chromosomes are replicated. This preparation is crucial for the subsequent stages of meiosis, ensuring that each daughter cell receives the correct genetic material. Interphase sets the foundation for the complex process of gamete formation. During prophase I, homologous chromosomes pair up and exchange genetic material through crossing over. This stage is visually striking under the microscope as chromosomes condense and become visible. The genetic shuffling that occurs here is a key driver of diversity in sexually reproducing populations. In metaphase I, tetrads, pairs of homologous chromosomes, align randomly at the cell's equator. This random arrangement is another source of genetic variation as it determines which chromosomes end up in each gamete. The stage is critical for ensuring the proper distribution of genetic material. During anaphase I, homologous chromosomes are pulled apart to opposite poles of the cell. This separation reduces the chromosome number by half, a defining feature of meiosis. The process ensures that each resulting cell will have only one set of chromosomes, ready for the next division. Telophase I marks the end of the first meiotic division. The cell divides into two haploid cells, each containing half the original chromosome number. This stage sets the stage for meiosis II, where the sister chromatids will finally be separated, completing the process of gamete formation. Meiosis II is similar to mitosis, but it starts with haploid cells. Sister chromatids are separated, resulting in four genetically unique haploid cells. This final step is essential for producing gametes that can participate in fertilization, ensuring genetic diversity in the next generation. Visualizing meiosis helps solidify our understanding of this complex process. Watching the stages unfold, from chromosome replication to the final separation of chromatids, demonstrates how genetic material is shuffled and distributed. This animation brings to life the intricate choreography of cell division that underpins sexual reproduction. Male reproductive system includes the scrotum, testes, and a network of ducts and glands. The scrotum is a sac of skin that houses the testes, which are responsible for sperm production. Each testis is divided into lobules containing seminiferous tubules, the site of sperm formation. Sperm travel from these tubules through a series of ducts, eventually reaching the epididymis, where they are stored until ejaculation. Testes receive blood from the testicular arteries, which branch off the abdominal aorta. Testicular veins form a network called the pampiniform plexus, which surrounds the arteries and helps cool the blood before it reaches the testes. This temperature regulation is vital for healthy sperm production. The spermatic cord contains these vessels, along with nerves and lymphatics, supporting the function and health of the testes. Sperm mature in the epididymis, which has a large surface area for absorbing excess fluid and passing nutrients to sperm. The ductus deferens or vas deferens is a muscular tube that propels sperm from the epididymis to the urethra. The urethra serves a dual purpose, conveying both urine and semen at different times, and is divided into prostatic, membranous, and spongy regions. Seminal glands or seminal vesicles contain smooth muscle that contracts during ejaculation. They produce a viscous, alkaline fluid rich in fructose, citric acid, enzymes, and prostaglandins. This fluid makes up about 70% of semen volume and provides energy for sperm, as well as substances that enhance sperm motility and fertilization capability. Prostate gland surrounds the urethra and secretes a milky, slightly acidic fluid that contains citrate, enzymes, and prostate-specific antigen, PSA. This secretion plays a key role in activating sperm and makes up about 1/3 of semen volume. The prostate's smooth muscle contracts during ejaculation, helping to propel semen through the urethra. Bulbourethral glands produce a thick, clear mucus during sexual arousal. This mucus lubricates the gland's penis and neutralizes traces of acidic urine in the urethra, creating a safer environment for sperm during ejaculation. These glands play a supportive but essential role in reproductive health. Sagittal view of the male reproductive organs provides a clear picture of how all the structures are arranged. From the scrotum and testes to the ducts and glands, each component is positioned to support the production, maturation, and delivery of sperm. This anatomical perspective helps us appreciate the complexity and efficiency of the male reproductive system. Relationship between the testis, scrotum, and spermatic cord is vital for reproductive function. The scrotum acts as a climate control system, keeping the testes at the optimal temperature for sperm production. The spermatic cord contains blood vessels, nerves, and lymphatics, all working together to support testicular health and function. Examining the male reproductive structures and the perineum gives us a comprehensive view of the organs involved in reproduction. The perineum, located between the pubic symphysis and the coccyx, supports the external genitalia and plays a role in sexual function and continence. Semen is a milky white mixture of sperm and secretions from accessory glands. Each ejaculation releases 2.5 ml of semen, containing 2,150 million sperm per milliliter. Semen provides nutrients, protects and activates sperm, and facilitates their movement. Its alkaline nature neutralizes the acidity of the male urethra and female vagina, enhancing sperm motility and survival. Semen contains prostaglandins that decrease cervical mucus viscosity and stimulate uterine contractions, helping sperm reach the egg. It also includes relaxin and enzymes that enhance sperm motility, ATP for energy, and antibiotic chemicals to destroy bacteria. Clotting factors initially coagulate semen, preventing it from draining out before it liquefies to allow sperm to continue their journey. Erection is the enlargement and stiffening of the penis, triggered by sexual excitement. Parasympathetic neurons release nitric oxide, causing smooth muscle relaxation and dilation of arterioles. Blood fills the corpora cavernosa, leading to engorgement and rigidity. The corpus spongiosum keeps the urethra open, allowing for ejaculation. Erection can be influenced by emotions and mental activity as well as physical stimuli. Ejaculation is the propulsion of semen from the male duct system. It is initiated by a spinal reflex over sympathetic nerves, causing the ductus deferens, prostate, and seminal glands to contract. The bladder's internal sphincter closes to prevent urine mixing. Semen in the urethra triggers muscle contractions, expelling semen at high speed. This event, known as climax or orgasm, is accompanied by increased heart rate and blood pressure. After ejaculation, the body enters a resolution phase, characterized by muscular and psychological relaxation. Blood flow to the penis decreases as arteries constrict, and the penis returns to its flaccid state. The refractory period follows, during which another erection or orgasm is temporarily impossible. This period lengthens with age, reflecting changes in physiological response over time. Erectile dysfunction, ED, occurs when there is insufficient release of nitric oxide, preventing erection. Temporary ED can result from psychological factors, alcohol, or certain medications. Chronic ED is often linked to vascular or nervous system issues, such as diabetes, atherosclerosis, or nerve damage. New medications like sildenafil Viagra enhance the effects of nitric oxide, offering effective treatment options. Spermatogenesis is the process of forming male gametes, or sperm, in the seminiferous tubules. It begins at puberty and continues throughout life, with adult males producing around 90 million sperm daily. This process involves several steps, including mitosis, meiosis, and the transformation of spermatids into mature sperm. Seminiferous tubules are lined with a thick epithelium containing 4 key cell types: cystenocytes, supporting cells; spermatogenic cells, which develop into sperm-mide cells which help move sperm and fluid; and interstitial endocrine cells, Leydig cells, which produce androgens. Each cell type plays a specific role in supporting sperm development and hormone production. Spermatogenesis occurs in 3 main steps. First, mitosis of spermatogonia produces primary spermatocytes. Second, meiosis transforms these into secondary spermatocytes and then spermatids. Third, spermiogenesis streamlines spermatids into mature sperm. This process ensures a continuous supply of sperm for reproduction. Spermatogenesis begins with spermatogonia, stem cells in contact with the basal lamina. These cells divide by mitosis, producing type A daughter cells that remain as stem cells and type B cells that move toward the lumen to become primary spermatocytes. This division maintains the stem cell pool while generating cells destined for sperm development. Primary spermatocytes undergo meiosis I to form 2 secondary spermatocytes, each with half the original chromosome number. These then rapidly undergo meiosis II, producing spermatids, small, round cells with large nuclei. This reduction in chromosome number is essential for producing haploid gametes. Spermiogenesis is the final stage, where spermatids undergo a transformation to become mature sperm. They elongate, shed excess cytoplasm, and form a tail. The mature sperm consists of a head containing genetic material and enzymes, a midpiece packed with mitochondria for energy, and a tail for movement. This process takes about 64–72 days, after which sperm are pushed into the epididymis to gain motility and fertilizing power.